All right, it's 9:02 here on the East Coast, and we have about 20 participants, so I think we're good to get started. So thank you to everyone for joining from all across the world, and welcome back to Vivo's webinar series, where we host a talk once a month focusing on topics relevant to 3D cell culture. Before we get started today, I wanted to mention Vivo's Win a ClinoStar 2 campaign. If you win, you will have the chance to work with a ClinoStar 2 for free for 1 year. The system will come fully equipped with the Vivo cloud service and the Hypoxia add-on unit. So please visit cellvivo.com or our LinkedIn page to learn more and enter to win. The last date of entry will be February 15th. Okay, so let's get started with our webinar today. So for this session of our webinar, we have Professor Krzysztof Wrzesinski, who will tell us the benefits of utilizing physiological oxygen levels for improved 3D cell culture outcomes. As always, before I introduce our speaker, I wanted to ask participants to please submit any questions you have into the chat function, and we will address them at the end of the talk. So to get started, I'll provide a brief introduction for Krzysztof. Krzysztof began his academic journey at the University of Southern Denmark, where he developed in vitro models for preclinical drug development. Since then, he has been heavily engaged in advancing this field, developing technologies for tissue mimetic cell culture. He is the co-founder and chief scientific officer of Vivo and the extraordinary professor at the Northwest University of South Africa. Please welcome Krzysztof.
Yes, thank you, Riham, for a very nice introduction. I would say, yes, in this webinar series, we will discuss a different aspect of the 3D cell culture and how those affect growth and maturation of the tissue, like structures, something like organoids and spheroids we are usually growing. So as Riham mentioned today, I will talk about how we can create a physiologically relevant environment for culturing our models. And I will talk a bit about the physiological hypoxia and how we can kind of recreate this situation in vitro models. So let's go to our slideshow. And then one thing which we should start with is like hypoxia is actually physiological state. It's characterized by a decreased oxygen level in organism and tissue compared to the ambient oxygen concentrations. Well, the oxygen is essential for the normal aerobic metabolism. A lower oxygen concentration influence a variety of cellular processes like cell growth, metabolism, as well as cellular development. And if we just look on this short, simple diagram, we can see that the levels of oxygen are very different in different parts of our body. When we are modeling in Vivo, we should actually provide an environment which is similar to what we can see in Vivo. Unfortunately, quite often we come to a halt based on the technological challenge where we cannot provide the right conditions for long enough time to get a tissue-like functionality. But before we go further to environment, I would like to just recapitulate on basic 3D cell culture keywords. So if we talk about 3D cell culture, we would use terms like organoids, where those are constructs created from primary or stem cells. We will use an word spheroid when we talk about the immortalized cell line construct. We will use term tumoroid, which is basically a construct made out of patient tumor cells. Or if we would say, would like to have something which is a result of a combination of two different organoids, that will be an assembloid. So all of them can be grown in the 3D, and when we grow them, we actually think about what type of environment we can provide to our cells, and we can use different models and different systems. And in basic, I would say that we could divide them into the two different categories: a static and dynamic culture. Where in static conditions, we use so-called passive diffusion. And when we use this as a nutrient delivery and metabolite removal, we actually create a very stable and mechanically gentle environment. However, creation of however, we will actually, by definition, create a diffusion depletion zone in such a conditions, and that zone will actually create a limitation for the how long we can culture and what size of the our construct we can achieve. To address this problem, quite often we introduce the media flow to kind of get rid of this diffusion depletion zone, and that's the way how we go from static to dynamic cultures. However, introduction of the media flow also introduce increased shear force, which affects our culture. And unfortunately, often when we use our technologies, we introduce shear force which exceeds many times any physiological relevant values of such a force. And then at Vivo, we have put a lot of effort to mitigate those technological shortcomings, which are actually kind of limiting the 3D cell culture models. We have actually tested our system in different setups, and if you are very interested in our test results, I would say I would very much encourage you to go to our webpage and look for quite a few publications we have accumulated there, which are describing the how we could use our system and how important is the conditions we are creating. But coming back to the technology, we have recently launched a new ClinoStar 2, which Riham was mentioning, that actually you can be lucky to get one for a year in your lab. But what is the ClinoStar 2 system? It's actually a new system based on a cloud control. So we can actually control, record and monitor all aspects of the culture via a cloud system, so it's remotely. The system is also having a capacity to regulate the oxygen levels, so create a more physiological conditions. The system is also using our standard disposable Klein reactors with individual humidification and gas exchange chambers. And those Klein reactors will actually come very handy with the preserving of the hypoxic conditions, for example. So, to those who see the system first time, I would say our system is utilizing a ClinoStar technology, where we are removing a depletion diffusion zone by using a gentle rotation of our vessel. And this rotation is removing the depletion zone, but as the vessel is completely filled with liquid, we are not creating turbulences, so we are not increasing the shear force. And we basically use the rotation to negate a gravity vector, or actually maybe to distribute, I should say, to distribute the gravity vector. But if I will show you this video now, you can see, as you could imagine, that you are inside the system. So we now actually on this video electronically removed the rotation of the vessel. So what you are actually seeing is only the movement of the constructs within the vessel. So they are actually staying in a very, very stable conditions. As a principle, Klein reactors and ClinoStar, you work it a bit differently than with standards plate-based systems. So actually, I am now showing you a video which shows the setup of ClinoStars in a real laboratory. So here you can easily see how nice and stable the constructs are behaving in the system. You can see actually different sizes, different ages of the construct. And yes, we do have our hypoxic system mounted. And here you can see us, our laboratory technician Kira is actually working with the vessel and how easy it is to actually work with it. The system contains several ports, and I would say that you could find out more on our webpage, how you use them, how you utilize them. It's a semi-closed cartridge-based system, which allows you direct imaging and control of your constructs. And obviously, it's as I mentioned before, cloud-based, so you can actually access your machines anywhere in the world. You can actually access and control it and record and adjust the parameters. So coming back to a system, and I would like to say that for us it's very important that this system is very stable and actually have a fast recovery. So as you can see on those graphs, we can provide a very stable environment, and the recovery is also very fast. Maybe I should say it here, depending when you open the doors, for example. So when you deplete the system out of the desired atmosphere, you can actually have a proper recovery with a temperature and CO2 within a very few minutes. As you can see, it's about 2 to 4 minutes, and you are completely recovered in the traditional way. What is very important is actually this green line here, because here's the experiment where we are opening the doors quite many times, and you can see the recovery of the CO2 and temperature. But what is important, when you measure those parameters inside the Kleino reactor vessels, you have a very, very stable environment. So actually, the cells in the Kleino reactors are not affected by the opening of our system. So we have been working on an hypoxia system. And here I could just show a glimpse on our functional model. So this is how it was, how it used to look like when we were actually working on and testing it. Right now, the hypoxia system is completely integrated into our cloud system. It's actually miniaturized, so it's much smaller unit, which you just click on the doors of the existing ClinoStar, and you control everything through the portal-based software. If you would like to see more about the system, please take a look on our webpage or just scan this code, and you will get more information about this. I just would like to show you a kind of most important things which people are asking quite often about. So the hypoxia system is able to create an environment from 2% to ambient oxygen levels. I would also say that if someone want to have a very, very low oxygen level, that's also possible. Chamber recovery time, which is very important. It's like if you have a setup of 12% of oxygen, you will get a 30 seconds recovery time after you have opened the system and basically exchange the atmosphere to a standard levels. If you will do the setup of 2%, you will have a 45 seconds or with less than 45 seconds of recovery time. And what is also very important that we use a very small amount of nitrogen gases to actually regulate the atmosphere. That is due to a special shape and size of the ClinoStar cell chamber. So if I just do this example that we have a 2% of oxygen setup, and we will do the 40 openings per 24 hours, you will basically use around 800 liters of nitrogen to regulate the atmosphere. So for some, maybe it's easier to see, okay, this would be around 16 bars on my 50-liter tank. That's for a current user of the ClinoStars and ClinoReactors. One other important factor is, okay, when we do this force gas regulation, we will utilize the humidity chamber in the ClinoReactors in a bit higher volume, but it's only around less than 8% higher. So actually it will not affect of use of the ClinoReactors. The important thing is that also when you use the ClinoReactors, as it is a semi-closed system, it actually allows you to preserve the hypoxic conditions for a short amount of time. Of course, it depends on the longevity of the time will depend on the how what are your setups. So the higher oxygen level, the longer time it will be preserved. And this will allow you, for example, to the imaging using a normal microscope without actually necessity of having a hypoxic chamber for your microscope. So if I would like to show you how fast it's actually recovering, or how actually fast actually we are able to stabilize environment inside the ClinoReactor when we are taking it from a normal atmospheric gas concentrations to a systems where we have an 2% setup. So it's actually equilibrating very fast, and it stays stable for a long time. And basically it stays stable. We can say that also opening the ClinoStar and a short exchange of the atmospheres are not affecting the environment inside the ClinoReactor. So with our system, with this new system, we still can use the same conglomeration method. So as I would say, we can still use the single cell suspension system. So this does not, the new ClinoStar 2 environment does not affect the abilities of how you will create the construct. So yes, this is a standard method where we use a single cell suspension and create a conglomerate. Of course, the micro pattern plates such as Kugelmeier, Agriwell, or Euplasia can be used to create an aggregate to grow them further in our ClinoStar or ClinoReactors. And I would say that this method is very nice as one of the way to create a controlled co-culture system. We also are able to use gels. So for those who use hydrogels, we say yes, it's completely possible to use it. We have tested gels based on sodium alginate, gels like Vitrogel. We also have used gels which are two-component based gels like Q-gels, and they all are working nice in our system. I would say that not so long ago, I was actually asked by one of our collaborators that can we support a different shapes than spheroids or them spherical shape. And the short answer is yes. This system is based on a very low shear force delivery of the nutrients. So actually we can support different shapes inside of the Clino clinoreactors. And here you have an examples of a shape created based on the microcarrier beads, but we also can use microscaffolds. Those are the actual electrospin fibers with shapes. So this is what would be kind of preserved and provide for the clinoreactors. Yes, so that was a short overview of how we can create, how the system look like, and how we create our models. And I would like to show you now a bit of research. So I have to say, yes, our Hypoxia unit is not very long on the market right now. We just launched it basically in end of last year. So in principle, from beginning of this year, we started. So we don't have a tons of the result yet, but we do have some collaborators which were testing our prototypes. And here, actually, I would like to showcase an experiment made by Anne Ager from Oslo University, from Jenny Rosalind group, where they were actually comparing structures of the fibroblast-based spheroids grown in standard conditions and in hypoxic conditions. And you can, it's not very difficult to see differences between the acetylate tubulin and actin structures in those constructs. So it's a very nice study. I think Anne will prepare this for a kind of publication very soon. And then another example is actually from a laboratory of Professor Claudia Bagni from Lausanne, where Giuseppe have been working on actually comparing quality of construct created in different methods, more traditional methods, as well as the ClinoStar. And he was actually comparing the reproducibility and all quality and level of differentiation of those constructs. And again, he has published this in a poster form, and I know that he's also preparing a publication from those data. I also would like to showcase a study which is already published, study from Romal Stewart from Australia, where he actually used the system to create a population of an neuronal construct or brain organoids, and those were used actually as a host for virus infections. So Romo was studying different versions of COVID virus and how they affect the neuronal tissue. So that's also a very interesting application. I also would like to show that it is possible to actually culture or keep alive patient biopsies in our system, and that is the ongoing study from Simone Sidoli group from Albert Einstein College of Medicine in New York. So they are actually using a ClinoStar 2 system, and they have a very nice results. I also hope that soon we will see some more out of this study. And another published data, this time from Dallas, from Texas, where Professor John Wu and Carlos Pinzo Arteaga have been using a ClinoStar technology to grow actually blastoids from bovine cells. And they actually constructed those from the induced pluripotent stem cells. And I would say they are very similar in structure as a native blastocyst. And that one is also a published study, so you can find much more in the publication. And yet another application where a group of Professor Doris Wilfingseeder has actually created a long organoid. So long tissue is often grown as an air-liquid interface systems, but here a group of Professor Doris Wilfingseeder has use and organoids, because they were also interested in studying of virus infections in a lung tissue, and this was also COVID-related studies. Yet another approach, which it's coming closer to me, as Riham mentioned, I am also an extraordinary professor at the Northwest University in South Africa. So here is actually a study from a group of Professor Krishna House, where the system was used to create a whole pipeline for a new drug development or studying of a new components which could become a drug, where in the system Professor Krishna's group has been creating and resistant and chemosensitive constructs to actually use it for the further tests of the new component. And there's another use of the system where actually Dr. Wien and Pfeiffer group has used the ClinoStar as an let's say a raw material production plant, because they actually work with a xenograft. So they use a construct created in ClinoStars to create and starting material for doing the xenograft and doing the study of the cancer in such a way. And a bit different approach, let's say for the end, this is actually coming from Slovenia, from National Institute of Biology and Professor Bojana Žegura group, where they actually are using ClinoStar. They use actually a lot of ClinoStar 1, and now they will be they will have a ClinoStar 2 as well, and they employ those machines to create a starting material to create a pipeline for genotoxicity tests. So that's also a very nice whole pipeline which Professor Žegura's group has developed, and it's a very, very impressive work they are doing there. So with this, I would like to say that I would like to summarize and finish the talk and say that those few examples of the technology and how it works and how it could be used will put will show you how versatile the system is, and I would say you probably could see how you could use this in your own research. But just to remember that what a ClinoStar is doing is basically creating the unique low shear force environment with high diffusion rates. It gives you ability to create a long cell culture conditions, and it gives you the conditions which mimic better in vivo functionality. And right now, we are also able to create a tissue-specific environment. So we are able to tailor the oxygen levels to a level which is existing in the in vivo situation. And with this, I would like to thank you for attention and for listening to me. And please do not hesitate to contact us if you have any questions. You always can find more information on our webpage, or basically just write to us, and we'll be more than happy to help. And as it was mentioned before, we have right now this special campaign. It's you can win a Cell Vivo ClinoStar 2 for a year to use in your scientific project. So thank you very much for listening to me. And yes, now I will give a voice back to Riham.
Thank you, Krzysztof, for your talk. So again, like Krzysztof said, you can win a ClinoStar 2 for free for a year. All you have to do is enter on our website or through our LinkedIn, and just keep in mind that the submission deadline is the 15th of February. So that's coming up soon. All right, so now we're going to move on to the question part of our talk. Krzysztof, I'll get started with some questions that have come in. So one of the questions is, what oxygen levels can be generated using the hypoxia system? Some tumor environments require less than 0.5%. Can that be achieved?
So yes, that's I'm trying to go back to my slide where what we do, it's like we have set up the default values on a 2% starting from 2%. That was default value. But as I said, our system is able to go to 0.1% or theoretically even to 0. But you will have to remember, okay, that will happen, but you will use more gases. So you will need to use more nitrogen to create such an environment. So and then you also have to take into consideration that what we are talking right now is a level of the oxygen, atmospheric oxygen inside the ClinoStar chamber. Your levels in the liquid where you have in the Clino reactors will be actually smaller. And that's what we have shown here, where you have a 0.5 if you set up your atmospheric levels to a 2% in the ClinoStar, you will get a 0.5% in your Clino reactor in your liquids. So that's so of course yes, we know that some cancer constructs actually really need to go low in oxygen, and yes, it's possible.
Great, thank you. The second question that has come in is, does it create a microgravity environment?
So that is the question which I very much like, and I would say, and unfortunately, no. And we have to say that what is a microgravity? It's an effect which you can achieve on the space station, or you can achieve it on the let's say a parabolic flight a bit. Our system works with 1G, but what it does, it creates a 1G around all directions, so all vectors. This is what the ClinoStat principle do. So you create around 1G and you distribute it all over. So you have a more like a normal conditions. And if you want to be extremely accurate, we say, okay, because we have a rotation, we actually have a bit more than 1G, but this a bit more is something after a third zero under the comma. So it's a very low additional values. So I would say we are doing we can simulate some aspect of microgravity. So that's how it should be called. Simulation of some aspect of microgravity. I know that people are using quite often the systems like this and call them microgravity systems. I have to admit that yes, I also published papers where we use a microgravity word, and we always said that it's simulated microgravity, but it should be simulated some aspects of microgravity.
Great, thank you, Krzysztof. Another question that has come in. Thank you for the nice presentation. What I haven't quite understood is the hypoxia unit already available for purchase? And
Yes.
If not, when can we expect it? I already have the ClinoStar 1, but would like the upgrade for the hypoxia unit.
So yes, that was how it was looking in our lab when we were testing. This is actually what you are now able to buy. Yes, you are able to buy the ClinoStar, the hypoxia unit. However, if this person already has a ClinoStar 1, so I would say you will need to have an upgrade to a ClinoStar 2 system, and yes, we are offering very favorable upgrades for ClinoStar 2. It's basically everything about the computing power of the ClinoStar 1 and ClinoStar 2. ClinoStar 1 was based on the Android-based communication, where the ClinoStar 2 is having his own cloud software, has a much higher computing power, so the videos and streaming, all those things are much nicer, and then the controlling of the gas and gas flows, it's basically possible. It would be, yeah. So I would say whoever has a class 1 machine should reach to our distributor or to us directly, and then we would definitely make it. But yes, it's on stock.
Great, perfect. Another final question. Based on the data you have shown, do you expect hypoxia to affect other cytoskeletal elements?
Oh, that's a question which I would say I don't know. I don't know. I would say, if you say, did you expect? I would say yes. I do expect quite a lot of changes when we do what we know from coming from the monolayer to the three-dimensional systems. We have seen how dramatic changes come to those cells, which we grow as a monolayer, or we grow them as in the 3D structure, so in the tissue-like systems. And we know that from our human physiology that's something which is really affecting the tissue, how it works. So I would say probably yes, but I don't have a data yet for that.
And Krzysztof, one question just came in. Can the CS2 record video for the time course of organoids?
So for right now, you will not be able to do so, and that's I would say, but that's definitely possible in the system. We have launched it right now, and when you launch new system, you always have to balance what new features will go to it. But actually, one of the big point why we decided to move to ClinoStar 2 from ClinoStar 1 was actually increasing of the computing power and ability of those new functionalities. And if you think about the recording, I can say right now it's not done, but those who have ClinoStar 2, they will get the update. And we also keep on upgrading the systems, and that's why the new button and the functionality will come to the cloud. I cannot say now when exactly, but not in far future.
All right, I think that is all in terms of the questions we have. Thank you everyone for joining us for the talk. I hope all your questions were answered. And I hope this was illuminating for you as it was for us. And Krzysztof, thank you for the talk.
Yeah, thank you very much, and thank you very much for having finding the time to listen to me. Thank you. Bye.
Thank you. Bye.